There is a molecule that millions of people have been told explains their mood or the absence of it. The theory is repeated in doctors' offices and popular books and everyday conversation, and it has been repeated for so long that it now carries the texture of a settled fact. Depression is what happens when the brain does not make enough of this molecule. A drug that restores it brings the chemistry back into balance. The molecule is called serotonin, and in 2022, a group of researchers led by psychiatrist Joanna Moncrief published the most comprehensive examination of the evidence for this idea ever assembled. They looked at 5 separate lines of evidence: serotonin levels and metabolites in the brain, the receptors that respond to serotonin, and the gene that controls how it is cleared from the synapse. They also examined studies in which serotonin was deliberately depleted and large genetic analyses. Across all 5, they found no consistent replicated evidence that depression is caused by a deficiency of serotonin. The review was published in Molecular Psychiatry, and the debate it triggered has not stopped. Some researchers pushed back, arguing the account was too narrow, or that the relevant evidence was more mixed than the review allowed. Those arguments are worth taking seriously. But the central finding has held. The story most widely told about serotonin was never as firmly supported as it was confidently repeated. What makes this genuinely strange is not that the theory was wrong. It is that the drugs built on it work. Antidepressants that target serotonin help real people feel measurably better, and they are among the most widely prescribed drugs in the world. The medication is not the myth. The explanation of how it helps is the part that turns out to be incomplete. That explanation is the one most widely given, the one most patients carry with them. Serotonin itself is a molecule of almost bewildering scope. There are 14 distinct types of receptor that respond to it. They are distributed across the brain, the gut, and the blood. It has been linked to mood and appetite and sleep. It affects pain, digestion, and the way an animal holds its position in a social hierarchy. Its most surprising feature, almost never mentioned in the popular account, is that roughly 90% of all the serotonin in your body is not in your brain at all. It is in your gut, doing something that has nothing to do with mood in any direct sense. The serotonin that does reach your brain seems to do something quieter and stranger than producing happiness. Something more like reading the state of the world and deciding when to hold still. How does a molecule most famous for its role in mood spend almost all of its existence in the digestive system? Why does a drug that changes serotonin chemistry within hours take weeks before it produces any benefit? And if low serotonin does not reliably cause depression, why does altering serotonin seem to matter at all? If you enjoy exploring questions like these, the ones where the popular account and the actual neuroscience have quietly diverged, alike would be highly appreciated. And if something here raises a thought or a question you want to put into words, the comments are there for exactly that. Now, let's begin. To understand what serotonin actually does, you have to start where it starts, inside the brain itself, in the small cluster of neurons where the molecule is first made. Deep in the brainstem at roughly the level where the brain meets the spine, there sits a collection of narrow scattered nuclei running along the midline. They are called the Raphe nuclei, from the Greek word for seam, because they are arranged like a biological ridge stitched down the centre of the brainstem. There are 9 of them in total, spread across different levels of the brainstem, and together they are responsible for producing almost all of the serotonin in the brain. The largest single source is the dorsal Raphe nucleus, which sits in the region just below the junction between the midbrain and the pons, tucked beneath the aqueduct of Sylvius. The dorsal Raphe alone contains around 235,000 neurons. That number may sound substantial. In context, it is tiny. The cerebral cortex alone contains something in the order of 16 billion neurons. The dorsal Raphe is an outpost, a population so small It would be unremarkable if scattered at random into the surrounding brain, and yet what it does is entirely out of proportion to its size. Before serotonin can be released by any of those neurons, it has to be built from scratch. The raw material is an amino acid called tryptophan, a molecule that the brain cannot make and that the body cannot assemble on its own. Tryptophan must come from outside, from protein in the food you eat. Animal and plant proteins alike are broken down in the gut and delivered to the bloodstream as their component amino acids. Tryptophan is one of them, but it is one of the larger amino acids, and to reach the brain from the bloodstream it must cross the blood-brain barrier using a transport system shared by several other large amino acids competing for the same passage. The amount of tryptophan that actually reaches a Raphe neuron depends partly on the composition of the meal that preceded it, and partly on the balance of other amino acids in circulation, a constraint that shapes serotonin production more intimately than most people realize. Once tryptophan reaches the brain and arrives inside a Raphe neuron, it undergoes two chemical conversions in sequence. In the first step, an enzyme called tryptophan hydroxylase attaches an oxygen atom to the tryptophan molecule and converts it into an intermediate called 5-hydroxytryptophan, or 5-HTP. Tryptophan hydroxylase is the rate-limiting step, the bottleneck that governs the speed of the whole production. A second enzyme then strips a carbon dioxide group from 5-HTP, and the result is serotonin. The finished molecule is packaged into small storage vessels inside the neuron called vesicles, held in reserve until an electrical signal arrives and triggers their release into the synaptic gap between neurons. After the serotonin is used, the brain breaks it down. An enzyme called monoamine oxidase, MAO, degrades serotonin inside the neuron after it is taken back up through the serotonin transporter. The breakdown product is excreted in urine, and early researchers used levels of this product in urine and cerebrospinal fluid as a proxy for serotonin activity in the brain. This is how several of the early studies of serotonin and depression were conducted, not by measuring serotonin in the brain directly, which was not technically possible, but by measuring the metabolic product that serotonin leaves behind. This indirect measurement would later become a source of complexity in interpreting what those studies actually showed. The actual naming of serotonin has its own strange history, and it begins not in the brain at all. In the 1930s, an Italian pharmacologist named Vittorio Erspemer was working with extracts from intestinal tissue and identified a substance that caused smooth muscle to contract. He called it enteramine, from the Latin for intestine. For years it existed in the literature as an intestinal compound, a curiosity of gut physiology. Then in 1948, a team of 3 researchers in Cleveland were investigating why blood serum caused blood vessel walls to tighten. Maurice Rapport, Ada Green, and Irvine Page isolated a substance from blood platelets that seemed to be responsible. They called it serotonin, from the Latin for serum and the Greek for tone, a molecule that tones the blood vessels. They believed they had found a cardiovascular regulator. Only later did it become clear that enteramine and serotonin were the same compound, the same molecule appearing in 2 different physiological contexts that nobody had yet connected. The molecule now known as serotonin had been discovered twice, in 2 different tissues, by researchers with entirely different questions in mind. And it would eventually turn out that those 2 different contexts, the gut and the blood, were hints about a molecule whose reach was going to prove far wider than either group had imagined. Here is the thing that changes everything about what serotonin can possibly mean as a signal. The dorsal Raphe nucleus sits in a fixed location deep in the brainstem, containing around 235,000 neurons. The brain as a whole contains roughly 86 billion neurons, a number that would take someone counting 1 per second more than 2,700 years to reach. The Raphe is a fraction of a fraction, and yet the serotonergic system originating in those tiny clusters has been described, by the researchers who spent decades mapping it, as having the largest and most complex outward projecting network in the human brain. The fibers that leave those Raphe neurons do not travel to one region or two. They thread their way into almost every major structure in the forebrain. The cortex and hippocampus receive serotonergic input. So does the amygdala and the thalamus. So does the reward circuitry driving motivation and action. There is almost nowhere in the forebrain that serotonin does not reach. The first researchers to see this clearly were two Swedish scientists, Annika Dahlström and Kjell Fuchs, working in the early 1960s with a new technique that made monoamine-producing neurons fluorescent under ultraviolet light for the first time. Using a chemical reaction that caused serotonin to glow alongside dopamine and norepinephrine, they were able to map the pathways these neurons projected through the brain. What they saw was startling. A small cluster of cells in the brainstem extended axons across distances that were, relative to the size of the cell bodies, almost implausible. A single Raphe neuron can branch as it travels, projecting simultaneously to multiple distant regions that serve entirely different functions, contacting thousands of target cells across the brain in a single sweep. Dahlström and Fuchs published their findings in 1964, and the map of the brain's monoamine projection systems they drew that year remains one of the foundational documents in modern neuroscience. Think about a single broadcast transmitter standing in one fixed location, one antenna on a tower, broadcasting at the right frequency. Every radio receiver within range picks up the signal regardless of what room it sits in, what function it serves, what it is tuned to receive. The transmitter does not know what each receiver is doing. It does not send different content to different addresses. It broadcasts a single state into the space, and everything that can receive it responds in whatever way its own design allows. This is what the Raphe does from the brainstem, a broadcast carrying across nearly every corner of the forebrain at once. This kind of architecture is unusual. Most neural communication is local, specific, directional. One neuron signals a defined target with a defined message. The glutamate system works this way, and so does the GABA system, the brain's main accelerator and main brake. A system that reaches everywhere simultaneously is doing something different. A signal that arrives everywhere at once cannot be carrying a targeted instruction. It is saying something about the overall state of the organism in the world, something about the conditions in which all the other signalling is taking place. And here is where the question sharpens, because the Raphe does not project uniformly to all regions equally. The dorsal Raphe and the median Raphe nucleus, the two main ascending sources, have somewhat different projection patterns. The dorsal Raphe tends to project more heavily to regions associated with emotional processing and motivated behavior. The median Raphe targets structures more involved in memory and the suppression of anxiety. These are not strict boundaries. The systems overlap and interact. But the point is that different subpopulations of Raphe neurons are reaching different functional territories, and within each territory the same molecule arrives at a different set of receptors. There are 14 distinct types of serotonin receptor, and they are not distributed uniformly. The receptor type determines what the signal means when it arrives. The same molecule released from the same source can slow activity in one region and increase it in another. The receptor type on the receiving end makes all the difference. A molecule that does this is not carrying a simple message. The architecture itself tells you what kind of signal serotonin is, and the architecture extends further than the forebrain alone. Raphe neurons also project downward into the cerebellum and into the spinal cord, where serotonin modulates pain processing and motor function. The molecule that is adjusting emotional response in the amygdala and memory consolidation in the hippocampus is simultaneously modulating pain signals travelling up through the dorsal horn of the spinal cord. One source, one molecule, reaching from the spinal cord to the prefrontal cortex. It is not a classical neurotransmitter in the sense of glutamate or GABA. It is something closer to a broadcast about the background conditions of the brain, and that raises the real question: what is it actually broadcasting? The brain already has neurotransmitters that deliver specific instructions. Glutamate tells a neuron to fire, GABA tells it to stop. Acetylcholine triggers muscle contraction and plays a fundamental role in attention and memory. These are messengers with a relatively defined signal and a relatively defined meaning at the receiving end. Serotonin does not work this way. It is not a command, it is a modifier, and this distinction is the key to understanding everything that follows. When a Raphe neuron releases serotonin into the synaptic gap, what arrives at the receiving neuron is not a message that says, "do this specific thing." What arrives is something more like a change in the receiving neuron's sensitivity, an adjustment to the threshold at which it will respond to all of its other inputs. Depending on the receptor type the receiving cell carries, serotonin may make that cell more likely to fire when stimulated, or less likely. It may slow the rate at which the cell responds to an incoming signal, or sharpen it. It may prolong the effect of another neurotransmitter, or cut it short. The effect is not fixed. It depends entirely on the state of the receiving neuron and the identity of the receptor it presents. Serotonin modulates. It adjusts the gain on the system it meets. There is a further distinction in how serotonin delivers this modulation, and it matters for understanding why its effects are so varied. Serotonin neurons fire in two modes. In one mode, they fire at a low steady rate regardless of what is happening in the environment. This tonic firing maintains a baseline level of serotonin across their target regions throughout the waking day, and is the background hum, the ambient serotonin that sets the general conditions for everything else. In the second mode, serotonin neurons fire in bursts in response to specific stimuli, particularly stimuli that are behaviorally significant or unexpected. This phasic firing delivers a concentrated surge of serotonin to specific regions at specific moments. The two modes do different things. The tonic background sets the overall sensitivity of the system. The phasic burst adjusts it in response to what just happened. Together they give serotonin a time resolution that a simple neuromodulator should not have. It is not just adjusting conditions, it is adjusting conditions dynamically in response to events. Think about the temperature setting in a large building. A thermostat does not tell any specific room what to do. It does not heat the kitchen and cool the bedroom independently. It adjusts the whole building's ambient conditions, the background against which everything happening in every room takes place. A room that was cold enough to make concentration difficult becomes more comfortable. A room that was too warm for sleep becomes tolerable. The thermostat did not change what was happening in any room directly. It changed the conditions under which those things were happening, and the rooms responded accordingly. Serotonin operates across the brain in a similar way. It adjusts the ambient conditions, the readiness and threshold of circuits in which every other kind of neural signaling is occurring. This is why serotonin appears in so many different biological functions. There is not a serotonin system for mood, and a separate one for appetite, and another for pain, and another for sleep. There is one serotonin system, and it adjusts the conditions of the whole brain. Mood changes when serotonin changes, so does appetite and pain sensitivity. Sleep, aggression, and impulse control all shift too. Not because serotonin is the molecule directly responsible for each of them, but because serotonin adjusts the background conditions of the systems that produce them. The 14 receptor types explain why the effect of this adjustment can look so different in different regions. The 5-HT1A receptor, when serotonin binds to it, tends to reduce the likelihood of the neuron firing it inhibits. The 5-HT2A receptor found in large numbers across the cortex, tends to increase excitability. It amplifies. The same serotonin molecule released from the same Raphe neuron arrives in a cortical region rich in 5-HT2A and increases activity there. It arrives in a different region dominated by 5-HT1A and quiets it. The signal is identical. The effect is opposite. The receptor is doing the translation, and with 14 types of receptor distributed differently across the brain, the same signal generates a different meaning in every location it reaches. This is what neuroscientists mean when they call serotonin a neuromodulator rather than a neurotransmitter. The distinction is not academic. A neurotransmitter delivers a message with a relatively fixed meaning at the receiving end. A neuromodulator adjusts the sensitivity of the receiving system to all of the other signals it is getting. It changes the background, not the foreground. Serotonin is not telling any region what to do. It is telling every region how responsive to be to what it is already receiving. What unifies all of those different regional effects is the modulating function itself, and that function turns out to have a consistent behavioral signature, one that took researchers a long time to recognize, because they were looking for it in the wrong place. They were looking at mood. The answer was in something older and more fundamental: how an animal holds its position in the world and decides when to move. The clearest window into what serotonin actually signals came not from studies of human mood, but from watching animals fight. In the late 1990s, a Harvard neuroscientist named Edward Kravitz had spent years studying how the brain governs behavior in lobsters, animals whose nervous systems are simpler than a mammal's, but share many of the same chemical building blocks. Lobsters establish dominance through combat. Two animals meet, posture, and fight until one withdraws. The loser shows characteristic behaviors of submission that persist long after the contest ends. Kravitz and his colleagues wanted to know what was happening in the nervous system during and after these encounters, and specifically what role serotonin played. The result was a finding that cut against almost everything the popular account of serotonin would have predicted. When serotonin was injected into a lobster that had just been defeated, one that had withdrawn in submission, the animal became willing to re-engage with the animal that had beaten it. It approached again. It displayed aggressive postures. It was prepared to contest a position it had just conceded. The serotonin had not made the lobster happy. It had made the lobster hold its ground. What changed was not the animal's emotional state in any simple sense, but its threshold for persistence, its readiness to stay and push rather than retreat and wait. The key finding: serotonin did not determine whether a lobster won. It determined how long the lobster was willing to fight. Dominant lobsters, Kravitz found, show higher serotonergic activity than subordinate ones. The correlation is not between serotonin and winning. It is between serotonin and the willingness to remain in contest. This pattern extends well beyond crustaceans. In primates, dominant individuals consistently show higher serotonin availability in certain brain regions than subordinate ones. When serotonin levels in primates are experimentally altered, social behavior shifts in predictable ways. Higher serotonin is associated with more assertive, persistent engagement with the social environment, with staying in the field and holding position. Lower serotonin is associated with quicker withdrawal, more reactive aggression, and reduced tolerance for frustration. What serotonin seems to be encoding is not pleasure, but something more like social confidence, the willingness to remain engaged rather than flee. In human research, the approach uses tryptophan depletion. Because serotonin is made from tryptophan, and the brain cannot store large reserves of either, reducing available tryptophan drops serotonin synthesis in the brain within hours. Participants given a low tryptophan regimen show measurable changes in behavior. Specifically, increases in what researchers call waiting impulsivity. The failure to defer action when a delayed reward would be the better outcome. The ability to hold off and stay the course, to tolerate a gap between desire and fulfillment, drops when serotonin drops. It is a small but consistent finding that points in the same direction as the lobster research, and the primate research, and the clinical observations about what happens to people when serotonin signaling is chronically disrupted. Think about what that amounts to as a description of this molecule's job. Serotonin does not seem to encode whether something feels good. It encodes the readiness to hold a position over time, to stay in the situation, to tolerate frustration, to resist the pull of immediate flight or immediate gratification in favour of persistence. When serotonin is active, the system leans toward inhibition of impulsive action and toward sustained engagement. When it falls, the system tilts toward faster, more reactive responses and lower tolerance for any kind of delay. The behavioural signature is consistent across species, across conditions, across experimental approaches. Serotonin says, hold. This is not happiness. It is something more fundamental than happiness, a calibration of the whole organism's relationship to time and to its position in the world around it. And this function is extraordinarily old. Lobsters have existed in something like their current form for hundreds of millions of years. The serotonin in their nervous systems does something recognizable in the serotonin of the human brain, not because the two systems are the same, but because the problem serotonin is solving is the same: when to hold and when to yield. What the molecule is measuring, across all of those species and all of those lineages, is not a subjective feeling. It is the readiness of the organism to stay where it is, which makes it all the more strange that this molecule became associated, in the popular mind, with happiness. That story has a history, and it begins not in the neuroscience but in a tuberculosis ward. In 1952, a group of tuberculosis patients at Seaview Hospital on Staten Island were enrolled in a trial of a new drug called iproniazid. The drug had been developed to fight the bacteria that causes tuberculosis, and in that regard it worked. In some patients, the bacteria retreated. But the medical staff noticed something else. Some of the patients on iproniazid began to seem unusually animated. They danced in the hallways. They laughed more. One physician described the ward as having an air of unusual liveliness. The drug was doing something to mood that had nothing to do with its intended target. When researchers investigated why, they found that iproniazid was blocking monoamine oxidase. This enzyme, abbreviated MAO, normally breaks down monoamine neurotransmitters including serotonin and norepinephrine. With MAO inhibited, these molecules were not being cleared from the synaptic gaps as quickly as usual. They were building up. The patients felt euphoric because, as far as anyone could determine, their monoamine levels were elevated. A class of drugs called monoamine oxidase inhibitors, MAOIs, was born from this observation. Some of them are still used today as antidepressants, and the mechanism is the same one that iproniazid stumbled onto at Seaview. Block the enzyme that destroys serotonin and norepinephrine, and the molecules stay in the synapse longer. Almost simultaneously, and from the opposite direction, another observation was arriving at the same hypothesis from the other side. Reuptake was a drug extracted from the root of a plant called Rauwolfia serpentina. It had been used in India for centuries to calm agitation. By the 1950s, it was prescribed in Western medicine to treat hypertension and sometimes psychosis. It worked by disrupting the vesicles inside neurons that store monoamines. Serotonin, norepinephrine, and dopamine leaked out and were destroyed rather than held in reserve for release. In a significant number of patients, Reuptake caused something that looked a great deal like depression. A flatness, a withdrawal, a loss of drive that disappeared when the drug was stopped. The reasoning that followed seemed almost too clean. Depression correlates with low monoamines. Reuptake depleted them, and produce depression. Euphoria correlates with high monoamines. Iproniazid increased them and produced the opposite. Therefore, depression must be caused by a deficiency of monoamines, and treatments that restore them should reverse it. In 1965, a young psychiatrist at Harvard named Joseph Schildkraut gathered this reasoning into a formal hypothesis and published it in the American Journal of Psychiatry under the title The Catecholamine Hypothesis of Affective Disorders. It was focused primarily on norepinephrine, but the same logic extended naturally to serotonin. Within the research community, serotonin became the version of the hypothesis that most captured attention, in part because the drugs most successfully developed from it targeted serotonin specifically. The hypothesis was extraordinarily productive for its time. It gave psychiatry a biochemical framework for thinking about depression at a moment when the field desperately needed one. It organized research in a useful direction. It pointed toward a class of drug targets that eventually led to the development of the selective serotonin reuptake inhibitors, SSRIs, in the 1970s and 1980s. Those drugs would go on to be prescribed to hundreds of millions of people. Fluoxetine, sold under the name Prozac, was approved by the FDA in 1987 and became one of the best-selling drugs of the 20th century. It gave patients and doctors a shared language for talking about a condition that had previously been described almost entirely in psychological terms. You have a chemical imbalance. This pill corrects it. The story had the appeal of all good stories. It was simple, it was mechanistic, and it put something complex and frightening into a form that felt manageable. The problem was that the story arrived before all of the evidence needed to support it was actually in hand. What was known in 1965 was that two drugs, one that elevated monoamines and one that depleted them, had opposing effects on mood in some patients. What was inferred from that was a general causal mechanism for depression. An inference from a handful of pharmacological observations to a general theory of how a major psychiatric illness works is a very long leap. The clinical observations were real. The hypothesis built on them moved much further than the data warranted, and for decades, because the drugs that targeted the system helped many people, the question of whether the underlying theory was correct got less scrutiny than it deserved. There is a particular kind of reasoning error in which a treatment's apparent effectiveness is taken as confirmation of the theory behind it. If the drug works, the theory must be right. But treatments can work for reasons other than the ones proposed, and the correctness of a mechanism cannot be read directly from the success of an intervention built on it. This would eventually become the central problem with the chemical imbalance account of serotonin. A theory is not tested by whether the drugs it produces help people. It is tested by whether the specific mechanism it proposes holds up when examined directly. Researchers began examining the serotonin hypothesis directly, measuring serotonin in the brains of depressed people and comparing it to the brains of healthy people. They asked what happens when serotonin is chemically removed. The results were disappointing in a very specific way. They did not find the consistent signal the theory predicted. The most direct test is tryptophan depletion. Give a person a drink that floods the bloodstream with competing amino acids. Tryptophan loses access to the brain, serotonin synthesis drops, and mood is measured. If low serotonin causes depression, this should reliably produce depressive symptoms in healthy volunteers within hours. In most studies, it does not. Healthy people who undergo tryptophan depletion feel slightly off, slightly flat, sometimes mildly irritable. They do not become depressed in any clinically meaningful sense. The finding that does hold up is smaller and more specific. Tryptophan depletion worsens mood in people who already have depression, or in people with a personal or family history of it. In those with no such history, the effect is minimal. The molecule seems to matter most to people whose systems are already under strain, which is a very different claim from saying that serotonin deficiency is what causes depression in the first place. Studies measuring serotonin directly have also failed to consistently show what the theory requires. 2 meta-analyses covering 19 separate studies of serotonin metabolites in cerebrospinal fluid, the closest available proxy for what is happening in the brain, found no consistent relationship with depression. Studies looking at serotonin receptor density found weak and inconsistent differences between depressed and healthy participants. These are not peripheral findings. Measuring the thing a theory says should be abnormal and consistently failing to find that abnormality is the kind of evidence that tends to end theories, or at least substantially revise them. Then there was the genetics. A polymorphism in the serotonin transporter gene called 5-HTTLPR was proposed as the inherited link, the genetic variant that made some people's serotonin systems less efficient and therefore more vulnerable to depression. A high-profile study in 2003 by Avshalom Caspi and colleagues suggested that people carrying one form of this variant were more likely to develop depression after stressful life events than people carrying another form. The gene-environment interaction seemed to confirm what the theory needed. The finding was widely cited and enthusiastically reported. Then large-scale replication attempts arrived. In 2009, a meta-analysis by Neil Risch and colleagues examined this interaction across many studies involving more than 14,000 participants and found no reliable association between the 5-HTTLPR variant and depression risk, either on its own or in combination with stress. The original finding did not hold at scale. These were not casual findings from small or poorly run studies. They were the results of serious, sustained research by people who expected to find what the theory predicted, and instead did not. The Moncrief Review of 2022 brought these threads together across 5 domains of evidence, and reached the same conclusion. No consistent, replicated evidence that depression is associated with lower serotonin activity or availability. The 5 domains included serotonin metabolites in blood and cerebrospinal fluid. They also included serotonin receptor binding, the serotonin transporter, and genetic studies. Across all 5, the pattern was the same, either no difference between depressed and healthy groups, or differences that were small and inconsistent across studies. The scientific response was mixed. Some researchers pointed out that the serotonin hypothesis had already been quietly set aside in serious academic circles years earlier, that the review was addressing a version of the theory no serious scientist still held. There is genuine truth in this. The research community had moved on. The problem was that the popular version had not. Patients were still being told by clinicians and leaflets accompanying medications that their depression was a chemical imbalance, and that their medication was correcting it by restoring serotonin to normal levels. The gap between what researchers understood and what the public was being told had become very wide. The situation was made stranger by a separate observation. SSRIs are also effective for anxiety disorders, for obsessive-compulsive disorder, for post-traumatic stress responses. These conditions are not usually described as states of depleted happiness. If the mechanism was specifically the correction of a serotonin deficit that causes depression, it would be surprising to find that the same mechanism relieves conditions that look very different from depression and carry no serotonin deficit story of their own. The drug's effectiveness across multiple diagnoses suggested that what they were doing was not correcting a condition-specific chemical fault. They were doing something more general. The theory was not wrong in the way a fraud is wrong. It was wrong in the way early scientific models often are wrong, built on observations that were real, constructed more quickly than the evidence warranted, and retained past its expiry because the drugs associated with it helped people, and because there was nothing cleaner to put in its place. What matters for understanding serotonin is what came after. The puzzle of the drugs that work by a mechanism nobody fully understands, and the search for what is actually happening in the weeks between the first dose and the first sign of improvement. SSRIs work by blocking a protein. The protein is called SERT, the serotonin transporter. And to understand the puzzle the drugs present, you need to understand what SERT does when nothing is interfering with it. When a Raphe neuron releases serotonin into the synaptic gap, the molecule crosses that gap and binds to receptors on the receiving neuron, producing whatever modulating effect the receptor type permits. But the gap also needs to be cleared. If serotonin stayed in the synapse indefinitely, it would overstimulate the receiving cell and disrupt the precision of the whole system. SERT handles the clearance. It is a protein embedded in the membrane of the sending neuron that binds to serotonin molecules still floating in the gap and draws them back inside, a recycling pump. The clearing happens fast. Most of the released serotonin gets pulled back within milliseconds and is either repackaged into vesicles for future release or broken down by MAO inside the cell. The synapse resets. SERT is not unique to serotonin neurons. It belongs to a family of monoamine transporters that includes the dopamine transporter and the norepinephrine transporter, each doing essentially the same recycling job for a different molecule. The chemical similarity between transporters is part of what made early antidepressants imprecise. Tricyclics blocked several of them at once. The development of selective reuptake inhibitors was an attempt to target only SERT, leaving the others alone, and that selectivity is where the S in SSRI comes from. SSRIs fit into the SERT binding site and prevent it from pulling serotonin back in. The result is that serotonin stays in the synaptic gap longer than it normally would, continuing to bind and rebind to receptors on the receiving cell, rather than being immediately cleared. This effect begins within hours of the first dose. Blood concentrations of the drug rise over days and stabilize within roughly a week depending on the compound. Fluoxetine has a long half-life and takes longer to stabilize than sertraline or escitalopram, but all of them are pharmacologically active within the first day. By the end of the first week, the SERT protein is consistently blocked during the hours when the drug is present. The serotonin signal in affected synapses is extended. The chemistry has changed, and here is the puzzle the simple theory cannot explain. If depression is caused by low serotonin and SSRIs raise synaptic serotonin within hours, depression should improve within hours. It does not. The therapeutic benefit, when it comes, typically begins to emerge after 2 to 4 weeks of consistent use. Some patients do not notice meaningful change until 6 weeks or beyond. The pharmacological event and the clinical outcome are separated by a gap that has never been satisfactorily explained by the serotonin deficit model. The gap cannot be explained by drug accumulation. Plasma levels of most SSRIs stabilize within a week. The delay is not the molecule building up to some threshold. Something else is happening in the weeks between the chemistry changing and the mood changing, and whatever that something is operates on a biological timescale that is much slower than the timescale of synaptic chemistry. This timing was noticed by clinicians long before anyone understood what it meant. Patients on iproniazid in the 1950s showed the same pattern. The enzyme was inhibited quickly, the mood lifted slowly. The phenomenon was real but inconvenient for the theory being built around it. A drug that works by correcting a chemical deficiency should work at roughly the speed at which that deficiency is corrected. A drug that produces changes in the physical architecture of the brain would work at the speed of biological growth, days and weeks, not hours. The weeks-long delay fits that second timescale far better than the first. Growing new connections between neurons takes weeks. Strengthening existing synapses takes weeks. Generating new cells in regions where cell populations have contracted takes weeks. Synaptic chemistry takes milliseconds. The gap between the pharmacological change and the clinical change is not an anomaly to be explained away. It is pointing at where the actual work is happening, and it is not happening at the level of serotonin in the synapse. The serotonin that SSRIs keep in the synaptic gap is almost certainly part of what drives the downstream process. The prolonged signal is probably the trigger, but if the trigger fires in the first hours and the response does not arrive for weeks, then the serotonin is not itself doing the corrective work. It is initiating something else, something that unfolds on the timescale of structural change rather than chemical change. What that something is took decades to begin to understand. The question the delay left open was not whether SSRIs worked. They did for many people much of the time. The question was what was actually happening during the weeks when the serotonin chemistry had changed, but the depression had not yet shifted. Something was being built. The evidence for what that something was began to emerge from a direction that had nothing to do with synaptic chemistry. Before structural repair can begin on a building, workers need somewhere to stand. The scaffolding goes up first. It is temporary, it is not the repair itself, but without it the repair cannot happen. BDNF, brain-derived neurotrophic factor, plays something like that role in the weeks after an SSRI changes the chemistry of the synapse. It is a protein the brain produces that promotes the growth and survival of neurons. It encourages the formation of new synaptic connections, supports the branching of dendrites, and in specific regions helps trigger the generation of entirely new neurons in the adult brain. The scaffolding makes the structural work reachable. BDNF is what the scaffolding is made of. The link between BDNF and depression was not obvious at first. What became clear through research in the 1990s and early 2000s, much of it from the laboratory of Ronald Duman at Yale, was that people with depression tend to have lower BDNF levels in certain brain regions. The hippocampus was the region that stood out. In chronic depression, the hippocampus can be measurably smaller than in healthy controls, not profoundly smaller, but detectably so, and the reduction correlates with the duration and severity of the illness, rather than being a fixed trait the person was born with. The structure is losing ground over time. BDNF is not the only neurotrophic factor involved in brain growth and maintenance. The brain produces a family of related proteins called neurotrophins, each with different regional distributions and receptor affinities. But BDNF is the most widely expressed of them in the adult brain. Its receptor is called TRKB. TRKB is found across the hippocampus, the prefrontal cortex, and the amygdala. These are the same regions that show structural changes in chronic depression. This spatial overlap between where BDNF acts and where depression leaves its physical mark is part of what made the neuroplasticity hypothesis compelling to researchers. The molecule that was reduced in depression and restored by antidepressants was acting precisely where the structural damage was occurring. The hippocampus was a particularly telling region to find this in. It is one of the few areas of the adult brain where neurogenesis, the formation of new neurons, had been definitively demonstrated. In 1998, Peter Eriksson and Fred Gage published evidence that new neurons are generated in the adult human hippocampus throughout life, using tissue from cancer patients who had been given a marker that labels dividing cells. The finding overturned a long-held assumption. Neurons, it had been taught, were fixed. You were born with the ones you had and losses were permanent. The hippocampus violated that assumption. New neurons were being made there continuously, and those new neurons were being integrated into the existing circuitry. What Duman and others found was that antidepressants increase BDNF expression in the hippocampus, and they do it on the timescale that matters. Weeks, not hours. The prolonged serotonin signal created by blocking SERT triggers a sequence of molecular events inside the postsynaptic neuron. That sequence eventually reaches the nucleus and activates genes that produce BDNF. The BDNF then promotes the growth of new connections and supports the survival of newly generated neurons in the hippocampus. The process takes weeks because growth takes weeks. Antidepressants that block adult neurogenesis in animal models, using radiation or other means, also lose much of their behavioral effect. The structural change is not a side consequence of the treatment, it appears to be part of what the treatment does. The neuroplasticity hypothesis that emerged from this work proposed that depression is not primarily a disorder of chemical levels, it is a disorder of structural adaptation. The brain under chronic stress loses connections in certain regions and fails to generate new neurons at the normal rate. The chemical changes associated with depression, including lower serotonin tone, may be contributing to that loss. But the repair is not achieved by raising the chemical. The repair is achieved by the structural work the chemical change eventually triggers. This reframing moved serotonin from a deficient substance to be replenished to a signal that initiates a slower and more complex process of physical change. The weeks-long delay in antidepressant response stopped being a puzzle to explain away and became instead a description of how long structural remodeling takes. SSRIs work where they work, not by filling a tank, but by starting a renovation. This serotonin holds the synaptic door open long enough for the renovation crews to arrive, which makes the distribution of serotonin in the body stranger to reckon with. If this molecule plays such a precise and consequential role in the brain's structural maintenance, what is it doing in the gut, where more than 90% of the body's serotonin is produced and never crosses into the brain at all? The fact that more than 90% of the body's serotonin never reaches the brain is not a footnote. It is the thing that most needs explaining, because a molecule this abundant in tissue, this remote from the brain, is not doing it by accident. The gut serotonin story actually predates the brain serotonin story. Vittorio Erspemer, an Italian pharmacologist, was studying the gastrointestinal lining in the 1930s when he found a substance being released by cells in the gut wall that caused smooth muscle to contract. He named it enteromin, from the Latin for intestine, and spent years characterizing its effects before anyone knew it was the same molecule that would later be isolated from blood serum and renamed serotonin. The gut had the molecule first. The brain version came to scientific attention later, and because the brain version arrived with a psychiatric story attached, it eclipsed what had been established in the gut. The cells that produce serotonin in the gut are called enterochromaffin cells. They sit embedded in the lining of the small intestine and the colon, interspersed among the epithelial cells that handle absorption. Enterochromaffin cells are sensory cells with a particular function. They detect mechanical pressure and chemical content in the gut lumen. When food arrives and the gut wall stretches under its weight, these cells respond by releasing serotonin into the tissue immediately surrounding them. That serotonin does not enter the bloodstream in a form that reaches the brain. The blood-brain barrier, a tight layer of cells lining the brain's capillaries, blocks serotonin from crossing into the central nervous system. The gut and the brain are operating separate serotonin pools that never directly mix. What the gut serotonin does locally is regulate the machinery of digestion. Peristalsis, the coordinated wave of muscular contraction that moves food through the intestine, depends on serotonin. When enterochromaffin cells release it into the gut wall, nearby neurons in the enteric nervous system detect it and trigger the downstream muscle contractions that push the contents forward. The speed and coordination of digestion is controlled in part by the rate at which enterochromaffin cells release serotonin in response to what is arriving. Drugs that block gut serotonin receptors can slow this process, and serotonin-related medications often list gastrointestinal side effects for exactly this reason. They are changing the chemistry that controls gut motility, whether or not that is the intention. But the gut serotonin does something else as well, and this is where the separate pools become relevant to the brain again. The vagus nerve runs from the brainstem down into the abdomen. Along the way it branches to the heart, the lungs, and the gut. It is the main cable connecting the central nervous system to the major organs of the chest and belly. It is often described as a two-way highway, but that framing misrepresents the proportions. Roughly 80 to 90% of the fibers in the vagus nerve carry information upward, from the body to the brain. The nerve is mostly a reporting line rather than a command line. Enterochromaffin cells sit in close contact with the terminals of vagal afferent neurons. When they release serotonin in response to what is in the gut, that serotonin acts on the nearby vagal terminals, generating signals that travel up through the vagus nerve to the brainstem. Those signals reach the nucleus of the solitary tract, a region in the brainstem that processes incoming information from the body's viscera, and from there they propagate to the hypothalamus and other regions involved in regulating bodily state. The brain is receiving a continuous upward report about what is happening in the gut, and serotonin is part of the language that report is written in. The signals that arrive via this route do not carry the message, "You are happy." They carry information about digestive load and nausea. They report satiety and the mechanical pressure of what is moving through the intestine. The gut serotonin system is not a mood system in disguise. It is a monitoring and coordination system that uses the same molecular vocabulary the brain uses for its own calibration, and it sends most of its output upward rather than downward. What research on gut bacteria has added to this is a further layer of the same story. The microbiome, the population of bacteria living in the intestine, plays a direct role in how much serotonin enterochromaffin cells produce. Certain bacterial species, particularly those in the Clostridia class, produce fatty acid byproducts of fermentation that stimulate enterochromaffin cells to increase serotonin production. Germ-free mice raised without any gut bacteria have substantially lower levels of serotonin in their intestines than normal mice, and their gut motility differs measurably from controls. The serotonin the gut makes is not just a product of the gut's own cells, it is partly a product of the ecosystem living inside it. Two serotonin systems operate in the same body using the same molecule. One is in the brain, adjusting the gain on neural circuits across cortex and subcortex, and calibrating responses to threat and social context. The other is in the gut, coordinating digestion and sending a continuous upward report about what is happening below the diaphragm. The molecule is the same. The functions are different enough to make it unclear why the same signal should serve both roles, other than the evolutionary tendency to reuse molecules that work. And there is one more use the body makes of this molecule, in a location that neither the brain nor the gut would lead you to expect. The pineal gland is a small, pine-cone-shaped structure buried in the middle of the brain, roughly at the point where the two hemispheres meet at the back. In adults, it is about the size of a grain of rice. René Descartes called it the seat of the soul, the point where the immaterial mind connects to the material body, in part because it is one of the few brain structures that sits in the midline without being duplicated on both sides. He was wrong about what it does, but he was right that it occupies an unusual position in the brain's geography. And it is where serotonin is converted into something the rest of the brain never sees. Every morning as daylight reaches the eyes, the retina sends signals along a pathway that eventually reaches the pineal gland and suppresses its main chemical output. Every evening, as darkness falls and that light signal lifts, the pineal gland begins producing melatonin. Melatonin is the hormone that signals night and promotes the onset of sleep, and the substrate for that melatonin is serotonin. The pineal gland does not synthesize melatonin from scratch. It takes the serotonin already present in the gland and converts it through a two-step process. First, serotonin N-acetyltransferase converts serotonin into an intermediate molecule. This enzyme's activity is controlled directly by the light signal from the retina. It rises sharply in the dark and falls back when light arrives. A second enzyme then converts the intermediate into melatonin. The result is that melatonin output from the pineal tracts the darkness with remarkable precision. It rises within a couple of hours of nightfall and falls back near dawn. The discovery of melatonin belongs to a physician named Aaron Lerner, working at Yale in the 1950s. Lerner had become interested in a decades-old observation that pineal gland extracts cause frog skin to lighten by concentrating melanin in skin cells, and he set out to isolate the active compound. The effort was not small. Over 4 years, his team processed enormous quantities of bovine pineal glands collected from slaughterhouses. From all of that tissue, they isolated a vanishingly small quantity of the active compound and published its structure in 1958. They named it melatonin for its effect on melanin and its origin in the pineal gland. In frogs, the skin-lightening effect is real. In humans, the relevant effect is the hormone's role in regulating sleep and circadian timing, which Lerner only began to explore after the isolation itself. What this reveals about serotonin is a function that has nothing to do with mood, social behavior, or the calibration of neural circuits. The same molecule that the Raphe nuclei distribute across the brain to adjust thresholds and set the gain on incoming signals serves, in the pineal gland, as the raw material for a timing signal that regulates the whole body's sleep-wake cycle. Serotonin is used twice, once as a neuromodulator acting during waking hours, and once as a chemical precursor transformed at night into the signal for sleep. This dual use creates a relationship between the two systems. Serotonin and melatonin are not simply two separate outputs, they are linked across the day. During the day, when light keeps the converting enzyme inactive, serotonin accumulates in the pineal gland. As darkness falls and the enzyme activates, that stored serotonin is drawn down to produce melatonin. The daytime serotonin is the reserve that nighttime melatonin draws from. The molecule does not move between the brain's Raphe circuits and the pineal directly, because serotonin cannot cross compartments freely. But the principle is the same. Serotonin during light becomes melatonin at dark. The practical consequence is that anything disrupting serotonin availability can have downstream effects on melatonin production. The same tryptophan depletion technique that failed to produce depression in healthy people does reduce melatonin output. The pineal gland has less serotonin substrate to work with when tryptophan is scarce. Light exposure at night suppresses the converting enzyme, reducing melatonin even when serotonin is available. And drugs that block serotonin's clearance from the synapse may also over time change the amount of serotonin available at the pineal. The same molecule manages social calibration in the ventral striatum and adjusts threat detection in the amygdala. It coordinates digestion in the gut lining and sends reports through the vagus nerve about what is happening below the diaphragm. At night, in the pineal gland, it becomes the substrate for the hormone that signals sleep. The connection to melatonin also helps explain why the pineal gland was so important historically for understanding the brain's relationship to the outside world. It is one of the few brain structures positioned to receive direct information about ambient light. The pathway runs from the retina along a dedicated track called the retinohypothalamic tract into the suprachiasmatic nucleus of the hypothalamus, which is the brain's master clock. From there, signals pass through a relay in the hypothalamus and down the spinal cord to the superior cervical ganglion, and then back up to the pineal. This indirect route, from eye to hypothalamus to spinal cord and back up to the skull, is the path by which darkness reaches the pineal gland and starts the conversion of serotonin into melatonin. The molecule that manages moment-to-moment calibration during the day is also the bridge through which the brain tracks the position of the Earth relative to the Sun. No single concept encompasses all of those functions, but the deepest one, running underneath all of the others, might be a simpler idea. Think back to the pill, the small white tablet. The idea that inside it a molecule will lift your mood because your brain has too little of something called happiness. That framing was never an accurate description of what the molecule does. It was a story that arrived before the evidence, caught on before it could be tested, and has proven stubbornly resistant to revision even as the evidence that should revise it accumulated over decades. Serotonin does not produce happiness. It does not carry the content of experience. What it appears to do across every system in which it operates is adjust the conditions under which experience is registered. It calibrates the threshold between stimulus and response. It shifts the brain's readiness to act, to persist, to stop. When the Raphe nuclei release it across the cortex and the limbic system, they are not delivering a message that says this is good. They are adjusting the sensitivity of the circuits that evaluate what is arriving. More serotonin in certain synapses makes a circuit more or less responsive to what comes next. Less serotonin changes those thresholds in the other direction. This is not a lesser function than generating happiness. It is a more fundamental one. A system that generates a fixed emotional state is fragile. A system that continuously calibrates the relationship between incoming information and the responses that information triggers is adaptive. What serotonin does is keep that calibration running, adjusting willingness to persist in the face of a social challenge, adjusting the gain on threat detection in the amygdala, adjusting the weight the prefrontal cortex places on immediate versus delayed consequence. These adjustments happen moment to moment, across circuits distributed through nearly the entire brain, from a cluster of cells in the brainstem smaller in total than a housefly. This may also be why the drugs that act on serotonin help conditions that have nothing to do with sadness. Social anxiety and obsessive-compulsive disorder and the re-experiencing of trauma are not versions of low mood, but they are all conditions in which a calibration is running in a way that does not match the actual demands of the environment. The threat response fires too easily, or cannot be quieted, or replays without a clear trigger. A molecule that adjusts the sensitivity of those systems would not need to target sadness specifically to shift those conditions. It would adjust the whole background, and the background is what those conditions are running on. In the gut, the same molecule is calibrating a different set of conditions. It detects what is arriving in the intestine and coordinates the muscular response that moves it through. It sends reports upward through the vagus nerve about what is happening below the diaphragm. It is not doing this as a byproduct of its brain function. It evolved to do this. The brain version may be the repurposing of a system that was doing gut coordination long before complex nervous systems existed, to calibrate social behavior and threat response. And in the pineal gland, serotonin is not calibrating anything. It is being consumed. In the growing dark, the enzyme that has been waiting through the daylight hours activates and begins converting the accumulated serotonin into melatonin. The signal for wakefulness and calibration is converted, molecule by molecule, into the signal for sleep. The same substance that has been adjusting your thresholds through the day is being disassembled to tell your body that the day is over. Right now, as you're lying here, that conversion is happening. Your Raphe neurons are firing more slowly than they were this afternoon. The serotonin that has been maintaining the tone of your waking circuits is beginning to retreat. The pineal gland is producing melatonin from the serotonin that accumulated during the day, and your body is reading that melatonin as the chemical statement of night. The molecule is not telling you how to feel, it is telling your brain when. That is the thing the pill story missed. Serotonin is not a signal about the content of experience, it is a signal about its timing and context. When to persist and when to yield. When to amplify a threat and when to soften it. When the day's calibration should give way to sleep. The molecule that populates greeting cards and wellness advertisements as the chemical of happiness is, more precisely, the chemical of when. And right now its message is unmistakable. If this kind of close attention to what the brain is actually doing is something you want more of, subscribing to this channel is the simplest way to find the next one when it arrives. The episodes are here when you're ready for them, and for now, the chemistry has already said what it needs to say.